to here's a quick look at what we'll be covering today. So we'll start by grounding everyone in the basics, what a bioprocess is and what we mean specifically by the upstream part of that process. From there, we'll look at some of the key challenges that come up in upstream biomanufacturing, especially when it comes to things like scalability, variability, and maintaining control over complex biological systems. And then we'll explore how digitization can help through 3 practical real-world examples. And finally, we'll end with a call to action. So now that we've outlined what we'll be covering today, you could just move to the next slide, James. Thank you. Let me briefly walk you through our objectives of this presentation. So our first objective is to introduce the core concepts of upstream bioprocessing and make sure everyone has a solid foundation. Then our second aim is to explore how digitization can add value through examples that highlight its impact. And finally, to offer some practical direction on how you might start applying these ideas in your own work or organization. So let's start with the basics. What exactly is bioprocessing? Simply put, bioprocessing is using living cells or parts of them to manufacture useful everyday products. You might be surprised to learn that bioprocessing is actually behind many common items, such as yogurt and cheese, which are made using helpful bacteria. Then we also have microbes, for example, which help produce biofuels, offering an alternative to traditional oil-based fuels, and enzymes, which are also made through bioprocessing and are found in things like detergents and even baking. Now, how exactly is this relevant to healthcare? Well, bioprocessing also allows us to grow the cells or proteins needed to make life-saving treatments. So, for example, it's how we produce insulin for people with diabetes, it's how we make antibodies that help treat conditions like cancer, and it's the foundation for advanced therapies, for example, cell and gene therapies that are shaping the future of medicine. So bioprocessing is a powerful way biology is harnessed to create products that improve our health and daily lives. Now that we understand what bioprocessing is and why it's so important, let's take a closer look at the key elements that make up a bioprocess. We're going from left to right, so starting with the process steps themselves. Bioprocessing isn't a single step, but rather a series of connected steps that transform living cells into a final therapeutic product. This process includes preparing the cells, growing them under carefully controlled conditions, harvesting the desired product, and then purifying and formulating it into the final treatment. Each of these stages plays a critical role and must be carefully managed to maintain the product's safety, quality, and effectiveness. Then we have collaboration, which is also vital. In a lot of cases, scientists and engineers work together to design and optimize these types of processes. So, for example, the scientists would focus on understanding the biology, while engineers develop systems and controls to make the processes reliable and scalable, which basically just means that it can be produced consistently in larger quantities without losing quality. Moving on to quality and control measures, which are integrated throughout the process. This basically just means that constant testing and monitoring is done throughout the process to detect any deviations or impurities. And these rigorous standards are essential to guarantee that every dose of the therapeutic product meets safety and efficacy requirements. Finally, we have innovation, which drives the field forward. So the implementation of new technologies and digital tools can help improve efficiency, reduce costs, and accelerate development timelines, making life-saving treatments more accessible to patients. Thank you. So to give a better overview of what a bioprocess may actually look like, we have an example right here on the screen. So a bioprocess is usually divided into 2 parts. We have upstream bioprocessing on the left-hand side and downstream bioprocessing on the right-hand side. Upstream processing is all about growing the living cells. So that means getting the cells ready, helping them grow, and making the product we want. Downstream processing happens after that, which is all about collecting the product, cleaning it up, and getting it ready to be used as medicine. Now, as we mentioned before, today our focus will be on upstream processing, so the first part of this. Next slide, please. Thank you. So upstream processing is the foundation of the entire bioprocess and is critical because the quality of the cells directly impacts the success of the final product. So here on the right-hand side, we have some of the main stages that are included in upstream processing, starting with choosing the right cells. We carefully select the cells that are best suited for producing the desired therapeutic product, whether that's a specific protein, antibody, or a gene. Then we grow the cells. So once we've selected our cell type, these cells are placed into bioreactors, which are essentially large controlled tanks, which are designed specifically to provide the perfect environment for cells to grow and multiply efficiently. After that, we feed the cells. So to keep the cells healthy and productive, they require the right nutrients, essentially food, food, oxygen, and a lot of other factors that are continuously supplied and carefully monitored throughout the growth phase. Now, to help visualize this, think of upstream processing like running a greenhouse. You start by picking the best seeds, in this case, the best cells. Then you provide them with the perfect mixture of water, nutrients, and warmth, which in bioprocessing terms means the right media and precise control in the bioreactor. And then once the cells are fully grown and ready, that's like harvesting your crop. So that then marks the transition from upstream to downstream processing, where the focus shifts to collecting and refining the product. Now, of course, upstream processing, like all parts of bioprocessing, comes with its own set of challenges. And these can vary depending on the type of therapy being produced or the skill you're working at. So some of these challenges are common across most bioprocesses, while others become much more significant when you're working with complex or advanced therapies. Now, I've put a couple of the main challenges here on the slide, and I'll work my way through all of them, starting with manual workflows in the pink box. So currently, many labs still rely on paper-based data recording, which slows down progress and increases the risk of human error. For example, you might have an operator which records bioreactor temperatures by hand. Now, this can lead to missed or incorrect entries, and if something were to go wrong, it's much more difficult to trace back what exactly happened. So as a result, the entire batch may be compromised or it needs to be discarded, which is not only time-consuming, but also incredibly expensive. Next, we have process inefficiencies. So without sufficient automation or coordination, tasks throughout a process are often repeated or delayed, which can reduce overall productivity and lower product input or output. Then we have tight process control. Small changes in conditions like temperature, pH, or oxygen levels can have a significant effect on cell health and productivity, making batch consistency quite difficult. So this can eventually lead to variable product quality or even failed batches, which again is a major risk in regulated environments like pharmaceuticals. Our next challenge is limited real-time visibility. Monitoring bioreactors in real time can be quite challenging, and without timely data, issues may not be detected early enough. Because of that, problems such as oxygen drops or contamination could go unnoticed until the next manual check, by which point some of the cells or even the entire batch may already be lost. Moving on to scaling difficulties. What works well at lab scale doesn't always translate directly to larger production volumes, making scale up a relatively complex task. For example, cells that grow efficiently in small flasks may not perform as well in large industrial tanks due to differences in, for example, mixing, oxygen transfer, or heat management. And this can then result in delays, rework, or even needing to redesign the entire process, which would obviously slow down development and again increase costs. Then another challenge is data silos and regulatory constraints. So data is often stored across many disconnected systems, and navigating compliance requirements can make access a lot more difficult. So this is a very fragmented approach, and it makes it a lot harder to make informed decisions. It also slows down troubleshooting, and it increases the risk of non-compliance with strict industry regulations. And then last but not least, we have process optimization during development. So a lot of the time, process decisions are based on the best results from a narrow set of tested conditions. But this doesn't always reflect the most scalable or robust solution. For example, if you'd be running a process at 3 degrees Celsius in small trials, that might give you great results. But maintaining that temperature at scale could be very energy intensive and hard to control. So, therefore, in these early stages, the decisions that are made need to be robust. Otherwise, scale up becomes a lot more difficult, which could potentially lead to delays, added costs, or again, failed batches further down the line. So these were some of the key challenges that we are currently facing in upstream processing. Now we're going to shift our focus and look at how digitization can help address some of these issues. And at this point, I'm going to hand it over to James, who will take us through that.
Brilliant. Thank you, Mirthe. So building on what Mirthe said about the challenges, during the development of any product or process that uses biology, there are distinct stages, all of which rely upon one another, and where the challenges that Mirthe has highlighted play a more or less prominent role. So if we start over on the left in discovery, where you're identifying your promising targets and organisms, your primary issues are in optimization, the sheer number of manual experiments that need to be run, and the volume of data that needs to be generated and managed as a result of that. And looking ahead, the ability, as Mirthe has already mentioned, to scale whatever conditions you land on as your preferred options for executing the process. So it might be really easy at a few milliliters, but you're talking about increasing the scale by tens, hundreds of thousands, even millions, up into the thousands of liters. So the whole environment changes, and as we've mentioned, what works at one end will not necessarily work at the other. Moving on into process development, you have your target and you're looking to develop the optimal process to be taken forward to commercial manufacturing. So your major issues are carried forward from discovery, but you've also added the real-time visibility of data because in effect, you're trying to create a small scale replica of the process that you'll ultimately be running. So knowing what's happening at any given time is incredibly useful. And then there's also the siloing of data. So between the systems or indeed the different teams that are involved, data needs to be brought together in order to learn from the process or experiment that you've run and move it on to the next iterative improvement. And finally, once you have a manufacturing process and you're running it at full scale, you then have to worry about controlling that process within the constraints that you've set, the regulations surrounding a product that's being made for human use, and of course, any inefficiencies that might be in the process, because anything suboptimal is creating an added cost in terms of finance, resources, time, and it's something that you don't want to bear. But fortunately, there are many ways in which digitization can help with these challenges, and we've highlighted just a few of the key areas here. So if we start over in data capture and integration by connecting the devices and systems and moving away from paper records, the capture of all data can be automated. And this results in a complete, permanent, trusted record of any experiment or process. It eliminates human error and is in effect the enabler for all other advantageous digitization options that follow. So there are a number of systems out there which support this at all different stages, from electronic lab notebooks to your lab information management systems, your manufacturing execution systems, and then the hardware devices that themselves, which can be connected to link the physical and digital worlds. Then you have centralized data and dashboarding. So here you're pulling all of that information, all of that data together. You're structuring it, contextualizing it, and making it available for insights and knowledge generation. So this can lead to your automated reporting and analysis, instant notification of any relevant process event, and ultimately the true optimization of a process because you have all of the data relevant to the process available at hand. So tools like cloud storage, data lakes, some of the more advanced visualizations options out there are all quite proficient at doing this. Then you have automation and process control. So whether that's physical or virtual, that can enable real-time feedback into a process. It will remove arduous manual tasks, and it holds the potential to hugely increase the throughput of your process. So you can generate more therapies for more patients at a lower cost, which is ultimately what this is all about, is the accessibility of these medicines to the people that need them. And then looking forward, we have predictive analytics and AI. So there are some tools available at the moment, but the potential of this technology would allow a true optimal process to be executed to the highest standard every time. But this requires knowledge, systems, and approaches that we just don't have yet. So the work being done in this area in the fields of process analytical technology, digital twins, where you have a virtual replica of your process and can run it indefinitely without actually having to execute a physical process, or indeed everyone's favorite buzzword, artificial intelligence, is incredibly exciting. So in order to help illustrate these concepts, we've prepared a series of analogies which are intended to kind of help relate the current position of digitization in upstream processing to ideas or technologies that you'll already be familiar with. So firstly, if we think about centralized data and dashboarding, we can draw a parallel to how navigation has developed over the years. So if you think back 15 or 20 years and you imagine a long distance car journey at this time, chances are there was an A to Z or a paper map involved. Someone in the passenger seat is acting as navigator. There's probably been a missed junction on the motorway. It's a lot of frustration. It's an incredibly stressful experience, and all round, it's just a very inefficient way to get from point A to point B. And sadly, this is where many processes, especially in cell and gene therapy, sit today. Very manual, very paper-based, very clunky, and hugely inefficient. This makes them very expensive, it makes them very laborious, and it makes it very difficult to get enough of them to the people that need them. So on the navigation side, if you fast forward a few years and the sat nav comes along, it's pulling data from satellites, it's overlaying your location, and it's providing you with a visualization of the route to take. It removes the need for the navigator, so that extra person who'll be checking your work. You have a greater confidence in the route that you're taking. But was it flawless? Absolutely not. Any one of us who had the pleasure of using a sat nav at the time would have experienced the pain of being led down a dead end road or being told by an autonomous voice to go cross country on a route that didn't appear to exist, or being stuck in a traffic jam that the system just wasn't aware of. And the same is true of many tools in bioprocessing now. It's pulling data from sensors. It can show you the response that it's taking, but it's hard to get a full appreciation of the journey because it doesn't have all of the information being pulled together in one place. And so we move forward to the current day and we all use our phone, we use Google Maps or Waze to get from point A to point B. It'll show you the best option for your route, depending on the mode of transport you want to take. It'll factor in traffic or engineering works, and it'll advise on the best alternatives given the current conditions. And this is the point that you want to aim for with a bioprocess. We know the start and end points, we know the constraints for getting there, but given the tools we have at hand, it's hard to use live information to help us get there in what is truly the most efficient way. So really, where most bioprocessors or tools sit at the moment outside of cell and gene therapy is between these sort of second two stages. So going from the data and control to the fully automated and optimized. Now, when it comes to automation and process control, we can think of a bioreactor like a home, only rather than a home for you and I, it's a home for millions or billions of the organisms that we're growing. And in days of old, there'd be a thermostat in the house, you'd adjust it to a desired temperature or an approximate desired temperature, and this would control your heating. And this is how the majority of bioreactor systems work. You have a sensor, in this case for temperature, you have a set point that it's controlling to, and you have a relatively simple feedback loop that keeps it as close to that as possible. But a bioreactor environment isn't that simple. There's a myriad of different conditions that you're trying to control to keep your cells happy and growing, many of which have an influence on the others, but they're not being controlled as though that's the case. And so they're all kind of competing to a certain extent. If you think of a slightly different example in terms of dissolved oxygen, there's a whole swathe of different influences on the bioreactor system that affect what is arguably the most essential thing for a living organism to exist and to grow in the form of the oxygen itself. So the temperature, the media composition, the gas composition, bubble size, gas flow rates, the mixing environment, all of these influence oxygen, but it will only ever really be controlled in the context of two of those readings that it has. And so you can think of the ideal state like a series of connected smart home devices. You can control your heating, lighting, TV, sound system, even your vacuum cleaner, all from a voice-activated device. And this is really what you want for the optimal control of your bioreactor environment. Rather than your classic PID loop, you have your temperature, dissolved oxygen, pH, all being controlled in the context of the others. And finally, if we look a little ahead at predictive analytics and performance, if you think of some of the more advanced smartwatch or smart ring technologies that are out there, measuring all kinds of different biometric stats, your heart rate, your O2 saturation, exercise, sleep, they're able to pull all of this data together and create a tailored recommendation for what you as an individual need in order to achieve peak performance. And this is what we want for our bioprocess. There are incredible advanced analytics available now, such as the example of Raman spectroscopy here, which can measure many different process variables at once. But the challenge is in processing that immense amount of raw data and translating it into useful insights that can be reacted to. This is partly because we don't have the same level of underlying understanding in our system as we do about human performance. And it's partly because the systems to utilize this data aren't fully developed yet. But this is what we're working towards, really. Real-time predictions of process performance and recommendations for optimizing this in flight. And nowhere is this more valuable than in cell and gene therapy. So just to round up, really, we've taken you on a very brief journey of upstream bioprocessing and the fundamental concepts, some of the places where you can apply digitization and hopefully help relate that to some more understandable things. And what remains really is for me to try and encourage you at whatever stage or situation you're at to start thinking about and employing digitization in your own work. It is incredibly powerful, incredibly valuable, and can only really help improve the experiments and processes that you're running at whatever stage you're at. So if you're at school right now, thinking about the experiments that you're running in the lab and the shortfalls that come with your paper notebooks and your lab reports and where digital tools might be able to help with that. If you're at university, starting to apply these tools in your projects, many of those that are available have free academic versions. And a word of kind of foresight, because of how powerful it is, and however you feel about it, digital is coming to biotechnology. By starting to use these tools now and become familiar with them, you're only preparing yourself well for an ultimate career in the field and in the industry of bioprocessing or biotechnology. And if you're in industry already, start to take a look around you at where the shortfalls are in, or the bottlenecks are in the work that you and your organization are doing, and where there may be tools either that we've mentioned here today or beyond that that could fill those gaps. Ways in which this tooling could help you make your life, your work, and that of your organization's operations better and more efficient. And so with that, I'll say thank you. I'll invite any questions, and I'll just add this is an area that Mirthe and I are incredibly passionate about. And so if there are any questions you have beyond the webinar, then please do feel free to reach out. We'd love to talk about it. Thank you. Okay. I'll stop sharing now, and I think we'll move into the questions area.
Great, thank you very much for that. Just to let you know, if you have any questions, please post them in the Q&A box. We're really open to taking those. I do want to read this out because it's a really nice thing. Somebody said it's fantastic and easy to grasp overview of bioprocessing, upscale, upstream, downstream, and very clear digitization info. They've really enjoyed that part of the webinar. So great, very kind words there already from the audience. But please, if there's anything you'd like to ask, please post it now. Bernice, is there anything that you'd like to ask them to expand on?
Yes, thank you. So I was interested to hear about the use of AI, and I know you said this is James. You said this is something that we talk about a lot now. And in terms of the, I suppose the next 5 years for the digitization of cell and gene therapies, where do you see AI coming in? Or what kind of developments do you see in the AI that we've, I suppose, already started using in digitization of that sector of the industry?
Yeah, so it's interesting. I was fortunate enough to be at a digital ATMP workshop at UCL a week or two ago. And this is something that came up there as well. And it's interesting, even the tools we use in everyday life, your chat GPTs or what have you, are being used today in order to write reports or SOPs for experiments that are happening in the lab right now. Even to kind of, generate the first drafts of documentation that really have historically taken weeks or beyond to prepare. So it's already in play and it's already being used. Where the challenge comes is people don't really understand the limitations, and so they will use it as a kind of as the way that they do in their everyday lives, right? Almost like a supplement for Google. And so we have to be aware of the fact that I think it's something like 30% of the time actually AI tools suffer from hallucinations. And so that's one aspect. It can't be regulated. There is no way of regulating a tool that pulls information from the entire internet. And so you need to find the right balance between having a useful tool that helps with efficiency and productivity versus becoming dependent on it. And so using that for the sort of initial drafting phase is one aspect, but not relying on it to basically do your job for you, because that's not what it's there for either, is where we kind of need to draw the line and have an appreciation and configure some policies for that. Sort of thinking beyond the documentation side, if we can start to build models around an understanding of the biology, which is tremendously complex, but we effectively have all of recorded human science and history to pull from, there is prospective tremendous value in the kind of models and virtual replicas of processes that you can build, and you can start to look at how to design organisms to be more effective and efficient, how to design processes optimally, and basically bypass a lot of the development pipeline. So a drug goes from taking 10 years to develop to, 1 or 2 years. It could really accelerate access to life-changing medicines.
Sorry, I think there are some questions. Thank you, James. That's really useful. And I think there are some questions in the chat. I mean, sorry, in the Q&A, not the chat. So if you can see them.
Yeah, I can actually see one of them. One of the questions is, what does a business associate do? And how did you get into this area after the biochemistry degree at UCL? So I guess that's a bit of a more career-oriented question. So while I was at UCL, I started working part-time at Exchange Data International as a business development associate. So what that entailed was following up on new leads, generating new leads. So basically just finding new clients that you can sell your product to, but it's less salesy and it's more focused on the strategy behind how you're going to find these people, how you're going to target them, and how you can help them. So then after I did that, that was more in the finance space. But obviously, as I was at UCL and I did my biochemical engineering degree, and then I did my master's, I wanted to make that transition from finance, obviously, to apply my scientific knowledge. So then I transferred to Autonomous, which is a company specialized in cell and gene therapy. So then I started applying my skills there. So that's how I transitioned into that space. But yeah, if you have any other questions, I'm happy to answer them also on LinkedIn if you want to send me a message. So
I think there's a second one here as well that says, were the degrees you've done relevant to the current positions you have? Were they necessary, or are there other alternative entry routes too? What's the most common entry path?
I think that really depends on the role you're going into. As I was specifically in a cell and gene therapy company, and as I obviously studied that at UCL as well, that was perfect for me because I had the base knowledge and then I was able to apply my business development knowledge that I had attained previously at my role before. So that was very helpful. But there was also a lot of people at the company who did not have scientific experience, and they had many other skills that they applied to the role. So it really depends on how the company operates, but I would say you don't need necessarily specific skills in order to actually start this. It would obviously help, and especially I think in actually attaining the role and in your interview processes and all of that, it is very useful, but you can always learn everything. So I think, yeah, it really depends. But as I really enjoy cell and gene therapies and biochemical engineering in general, for me, that was the clear path forward, and it definitely helped in my role.
Yeah, I'd probably just add to that as well. Beyond the kind of educational side, Mirthe and I both did biochemical engineering, but from there we kind of went two quite different routes to end up in a similar place. So Mirthe went business development first, and I went down a much more technical route, applying that to a number of different fields, and then worked my way back round to the partnerships and business development side. So there isn't really a one size fits all. I think most important is to ensure that you follow where you have the passion for the space, rather than trying to sort of retrofit your kind of education to a desired endpoint. There will be things that you find out on the way and opportunities that arise that will take you in a direction that you never really foresaw, or that's certainly what's happened to me and a lot of people that I know. And so yeah, there is a degree of planning you can do, but as is often the case, life gets in the way. So let it kind of take you where it best suits you.
Yeah, just on that, especially when it comes to business development, when you are selling a product and you're trying to help people, actually being passionate about it, what James said, and actually believing in it is a big help, because then people will actually be able to also see that and understand it. So I think, yeah, just adding on what James said, choosing the right path for you and your interests and following that, that is probably our biggest advice.
Great. Those were fantastic answers. And I think it really does help with the types of groups of people that we are trying to help, the types of students that we target with all of the materials and all of the resources that we have at Resilience, because we do offer many tools and many teaching materials and courses for vocational courses. And so I think it does help to have a practical understanding of the industry that you just gave today. That's very helpful, and it really complements the work that we do in the lab with these students. So, yeah. If anyone has from the audience, if anyone has any questions just about just digitization, the content of the webinar, that would be really useful, because I think the students who are accessing this webinar after it's been recorded, if we can show the link between what they're already doing and what you've talked about, I think that would help. I know we have some people from Brightest, and you work with helping people understand these types of processes. So.
Yeah, I think one of the really interesting parts for Mirthe and I in generating this session was thinking about, we were briefed on basically aim this at anybody from the age of 13 upwards, which is a huge spectrum to try and cover when you're talking about what is really quite a potentially complex space to dig into. And so that's where the sort of analogies come from in trying to relate, very complex technical ideas to something much more kind of bite-sized and understandable. So it was a really fun and interesting challenge for us to sort of come up with translating those concepts.
I do have one question related to just the skills, some of the skills that you've included that are I think if I give one example, just really understanding how to set up a bioreactor, and thinking about all of the parameters that you need to take into consideration, like oxygen tension, the types of media that you use, how you supplement those media to keep your cells alive, depending on the type of bioreactor that you've set up. So, so really thinking about this in terms of digitizing that a protocol, for example, a protocol for setting up a, let's say, a fixed bed bioreactor. Is this an example that you've come across, and have you been able to, do you have a protocol that you've digitized for setting up any type of bioreactor?
So there's there's two pieces to that question that I think would be interesting to touch on. As you're aware, there's the sort of virtual reality option for training in the setup of bioreactors and sort of establishing the protocols that way. The other piece I'd kind of touch on briefly is, way back when I was working, it's a simpler example than what you're talking about, but way back when I was working at Lonza in the pilot plant, and we used to, on a daily basis, have to take some measurements, go into an office, run an Excel basically program to figure out what our feed rate should be for the next 24 hours, come back to the lab and sort of change a calibrated pump to update that. And so you'd see the kind of fluctuations in nutrient concentrations jumping up and down. Now that's arcane, and you're looking at really sensitive organisms that, don't respond well to a fluctuation in conditions, and that's your standard practice. I'm sure it's moved on since then, but just the ability to take the information either in real or near real time and feed that directly back into the system to update a flow rate is, it's pretty much common practice in everywhere I've worked since. So, things move on in that regard. And so it kind of points to the point we were saying earlier. It's coming, it's happening. It's just how advanced we can get, how quickly, and keep up.
One colleague comment on the fact that we might not need people in the future to run these types of upstream and downstream processes, because if these systems are digitized and we use AI to, as you say, optimize our experiments and use previous data that you've generated to know that, okay, so if I have this level of growth of my yeast cells or my mammalian cells, I should modify the medium composition or the amount of medium or the oxygen in this way to get the yields that I need, or the yield of a particular product. And so you wouldn't need any input from people. And I don't know what, for both of you, this is a question for both of you. I don't know what you think. Say 10 years from now, there would be no need for people, because everything is run by AI.
I think that depends on the type of therapy you're manufacturing. When it comes to advanced therapies, especially looking at, for example, cell therapies, a lot of them are quite personalized. And in situations like that, you would still need people in the labs, because they are very, every therapy, every patient's cells are different. So they need to change the processes according to that. But in general, I think, I'm not sure about in the next 10 years, but in the future, at some point, I think we could move towards that. What do you think, James?
Yeah, I think it's the automation side has kind of happened in other industries, right? And so I would expect it to follow a similar trend. If you think about automotive manufacturing, that used to be an incredibly manual process and now is very, very heavily automated, but you still haven't removed people from the process entirely. It's much more sophisticated. The throughput's much higher. The reproducibility is much greater, but you haven't entirely removed people from the process. I think what's more likely to happen is it'll just be a shift in bottleneck, right? You'll use AI or automation in order to improve a particular problem that we have now, and that will lead to, it'll unblock another challenge. It'll just get pushed down the line to a certain extent. So people will adapt, they will upskill, they will retrain, and their specialities will shift slightly. I don't think there is a point in such an advanced field where we will be completely removed from the process. We'll just be doing slightly different jobs in 10 years to what we are now.
I think if we have no more questions or comments, and James and Mirthe, do you have any concluding remarks or anything that you want to just any key points you want to add before we finish?
I don't think so. Just to say thank you again for coming along. Reach out if you have any further questions, and thank you as well for the very kind words in the feedback there.
Great.
Yes, thank you, everyone. Also, if you would like to have the slides, I'm sure we can find a way to send them over to you. If you would want any further information or anything, let us know.
Thank you. That's very generous. Thank you, Mirthe.
Great. Thank you very much. Been great to have you with us. I'm going to call this over, but thank you very much. Have a great day and great week. Bye-bye.